So, welcome, Dr. Yan.
Thank you. Good morning, Council Chair, ladies and gentlemen. I am Helen Yan from the Department of Pathology, Hong Kong U. Because of Professor Leung's personal condition that she cannot be here physically today, and I will present on behalf of her on the project from a TRS project under her supervision. Stomach cancer remains as the 5th most common cancer type and the 4th leading cause of cancer death worldwide, and with exceptionally high incidence rates in Eastern Asia, including China and Hong Kong. And in year 2020, there were more than 1 million new cases and over 0.7 million deaths. In Eastern Asia, again, it has the highest mortality rate. The current challenge to treat gastric cancer patients include a high mortality rate, early dissemination, lack of biomarkers, and effective drug. There are couples of known risk factors that could increase the chance of getting stomach cancer, including family history, age, tobacco alcohol use, obesity, high salt diet, and infections with EBV and H. pylori. And all these factors can lead to DNA damage and cause mutations that accumulate in the normal stomach cells. With advances in next-generation sequencing technologies, cancer's genome has been widely studied, and stomach cancer is subdivided into different subtypes, and each subtype has their unique driver mutation, which we have summarized here on this circle plot, with the heterogeneous natures of gastric cancer that highlight the need for personalized medicines. The current treatment of stomach cancer patients is surgical resections, if possible, followed by administrations of cytotoxic chemotherapeutic agents, including 5-FU, cisplatin, epirubicin, or their combinations. Some patients show good response, some has no effect, and some even show harmful side effects. Instead, personalized treatment is what the current cancer patient is now all looking for. But to achieve the genome-guided personalized treatment therapy, we need to have deep biological understanding of individual driver and also their effect when they're in combinations. We also need to identify new vulnerabilities and deregulation pathways so that we can have new cancer drug development. However, there has been a lot of challenge in studying gastric cancer. Apart from the intrinsic complex molecular features of gastric cancer, there has been a lack of good and scalable preclinical model, a lack of normal gastric cell line to study driver gene functions, and we also need a complementary cell model which can bridge the mouse and human studies. However, until recently, most of the cancer studies were conducted using 2D cell line that originate mostly from advanced cancer stage, and they also have limited number. For instance, in gastric cancer, we only have 42 gastric cancer cell line commonly available in total in the world. That cannot represent all of the subtype of gastric cancer and cannot trace stepwise origin from normal to cancer. Moreover, the cell keep changing over time through accumulating various mutations. Therefore, our team has been establishing the 3D organoid cultures by adapting the protocol invented by our collaborators, Professor Hans Clifford, since 10 years ago, which we dissect a small piece of cancer and normal tissues from mutations. We perform cell isolations, put the cell in the matrigel, supplement with culture medium with a list of leach factors. Then the cells start to grow, expand, and divide and form a 3D cluster. And this is how the matrigel looks like under the culture dish. You can actually see the tiny dots of organoids by naked eye when they are big enough. And this is the one normal organoid that labeled with GFP that you can see under the microscope. And you can see actually see the individual cells are actively dividing. With the normal organoids, then we have the chance to study the functions of individual driver in mediating cancer, and also study their response towards different anti-cancer drugs. For instance, here we have the normal organoid that engineer with the SMAD4 and P53 mutation individually. And we already see the difference in terms of their cell cycle, in terms of their cell divisions. And upon drug treatment, you can see the SMAD4 mutant organoid respond well, and they die immediately, but this does not happen in the P53 mutant organoids. In our TBRS project, we have established more than 80 tumor organoids from 58 gastric cancer patients, cover most of the subtypes of gastric cancer. And we also perform really comprehensive molecular profiling on them, including the somatic mutation, copy number variation, gene expressions. We perform methylation array, and we began to look into the proteomic changes in the cancer organoid, as well as understand their intra-tumor heterogeneities through single-cell RNA sequencing. We also perform extensive functional characterizations on our organoids, including the cancer and some of the pair organoid, so as to document their growth rate, their revival rate, the niche factor dependence, the cell-cell adhesion dependence, their histology, whether they're present of the EBV genome for those EBV-positive cancer. And together with the long-term clinical follow-up data, these biobank represents a really huge resources that help us to understand functional biology of the biology of gastric cancer, and to have performed drug screening studies. Here are some of the representative H&E and bright field images of our cancer organoid from the biobank. For the normal organoid, it always look like a cystic with a spherical in shape, single cell layer with a large lumen. For cancer that carry EBV genome, generally have a cystic morphology, ruffled with multiple cell layers. Typical intestinal organoids show irregular in shape and with occasional formation of small lumens. For diffuse type GC organoids, they form a solid cell cluster, and with some show a loose cell aggregate, just like a grapes. And for the mucinous cancer organoid, you can actually see the secretions of mucus from the tumor cell in vitro. Here is the slide that's representative images of the organoid and the paired frozen tissue, and you can actually see that the cancer organoid look really similar to that of the cancer cell in vivo. Our cancer organoid also capture the regional heterogeneities of oncogenic protein expressions. For instance, these cancer patients that have the ERBB2 amplifications, and we can really see heterogeneous expression of HER2 proteins in the organoid as well as in the paired frozen tissue. Similarly, these patients that have the FGFR2 overexpressions, we can also see these heterogeneous expressions in both the cancer tissue as well as the organoids. In terms of the tumor purities, our cancer organoid is a lot higher than that of the paired frozen tissue, as indicated by the bar and the star here. By calculating the allelic fluctuations from the key driver and also from the facet analysis using the whole exome sequencing data. This is the oncoplot summarizing the somatic mutation profile of a subset of our gastric cancer organoids, and essentially they recapitulate those previously observed in gastric cancer patients, including an enrichment of P53 mutation in the intestinal type GC, frequent mutation of ARI1A in MSI and EBV positive cancer organoids, and we also found frequent CDH1 and deregulation of Rho pathway in the diffuse gastric cancer organoids. We also look at the somatic copy number alterations. And again, the tumor organoid recapitulate the in vivo chromosomal stabilities and instability patterns in different molecular subtypes of gastric cancers. Although both the normal and cancer organoid, they are cultured under the same conditions, receiving the same culture media, but they can still preserve their own distinct transcriptome profile and show differential pathway activations. For instance, the cancer organoid show an upregulation of intestinal signatures, cell cycle, Hox family gene, and Wnt signaling in some of the cancer organoids. For our MSI gastric cancer organoids, similar to their pair frozen tissue. We found a lot of SMV and indel. And we also reviewed the underlying molecular mechanism, either due to somatic promoter methylation of MNH1, with one case that we identify a germline MNH1 mutations. For the EBV positive gastric cancer organoids, we confirmed the presence of the EBV genome in our cancer organoids, with one that we have been cultured in vitro continuously for 6 months. We're still able to detect the presence of this EBV genome. Besides both the lytic and the latent genes that were found in the frozen tissue, we are again able to detect them in the cancer organoids. In our biobank, we have identified a few number of patients that carry this claudin 18R gap fusion transcript, representing the first in vitro model that's covered with these unique molecular alterations. Besides, we discovered another new isoform of RGAT26 called the delta catenin RGAT26 fusion transcript, and we confirmed the presence of these fusion proteins in our cancer organoid by means of Western blot. We also look at the tumor evolutions in our cancer organoids. With this river plot show how the clone evolved from the same patients. And this is the phylogenetic tree show how the individual clone evolved from the top. And in these particular cases, we established organoid from 2 regions. One is from the dysplasia at the tumor edge. Another is from the invasive cancer. And we can see that initially they share the early driver mutations, including APC and P53. But when they start to evolve, they tend to gain different kind of driver mutations. We also look at the drug response between patients on post-op drug treatments and their corresponding cancer organoids. For instance, these 2 cancer patients, after the gastrectomy, they were on the 5-FU and cisplatin, and we found a significant shrinkage on their tumor size. And in parallel, their cancer organoids, they also showed really good response on the treatment with the combination of 5-FU and cisplatin. However, for this patient, GX68, he was under 5-FU after surgery. However, we see a continuous enlargement of the liver metastasis, which consistently his cancer organoid does not respond to 5-FU in vitro. We also established a really large scale drug screening pipeline in this project by adapting the protocol invented by our collaborator in Sanger Institute, which we are able to perform drug screening with 37 drugs at 7 concentrations on 1 organoid within a week. And this pipeline involved 2 robotic systems, including one that helped us to seat the organoid in the cell at day 1, followed by another robotic system, the liquid handler, that helped us to dispense drug using the aquatic sounds energy. And we perform an endpoint analysis to look at the drug response in terms of cell viability at day 7. We are able to show the high reproducibilities of these of our drug screening pipeline, in which we perform the drug screening on the same organoid but at a different passage as biological replicates. And here you can see the really high correlation between biological replicates of all our tested organoids. The response to NUTNIN3A is always our positive control in our drug screening pipeline, which all the P53 mutant line were sensitive to NUTNIN3A as shown in this viability plot. And when we apply the elastic net regression model, we again see the P53 mutation as the top features that can able to predict the sensitivities of NUTNIN3A. And so far, we have finished drug screening on 66 tumor organoids and some of the normal organoids. And we are preparing the manuscript on this, and we hope to share some more of these findings to you or in the near future. This slide essentially summarizes the key applications of organoid technologies, and in this project, we have made use of normal organoids to identify new carcinogen exposure signatures, and we perform disease modeling through gene editing. And for the cancer organoid, we have established a relatively large gastric cancer biobank and make use of it for disease modeling and drug screening. Apart from the gastric cancer organoid, we also established a colorectal cancer biobank enriched in early onset patients that I will talk a bit more in the following slide, and also a few of the cancer organoids that originate from hepatocellular carcinoma, nasopharyngeal carcinoma, and the esophageal squamous carcinoma. This is the oncoblock summarizing the key somatic mutations that we identified from the colorectal cancer organoids. And they were arranged according to the patient age and divided into the early and the late onset. As you can see here, the late onset CRC patients, they always carry the APC mutation, whereas for the early onset patient, the chance of getting the APC mutation is less. And we subsequently identified the previously described PDP-RK, R-responding fusions, in 5 tumor organoids derived from 2 APC wild-type patients by RNA sequencing. And we subsequently confirmed these fusions by RT-PCR and Sanger sequencing, representing the first in vitro 3D model of these R-responding fusions. After that, we identified a RNF43 splicing event in other APC wild-type cancer organoids that we further confirmed by RT-PCR as well. And interestingly, in one of the cancer patients, age 15, that we find a substantial increase in both the number and densities of SMV, and in both the normal and the tumor organoids. And this patient is actually a cancer survivor from a chemotherapy for acute myeloid leukemia and received a cyclophosphamide and busulfide treatment for bone marrow transplant at her young age. In particular, we find a dominant C to A transitions in both the organoids and in their paired frozen tissue, whereas the C to T transition is commonly found in other CRC patients. And so after that, we find that this novel mutational signature can actually induce in vitro when we treat the normal organoid continuously for a relatively long period of time with a combination of drug called the gazinovir and a mycophenolate mofetil, which is exactly the combination of drug that the patient received for a prolonged period after bone marrow transplant for acute leukemia, suggesting that this mutation can be induced by GCV and further enhanced by MMF. Despite the difficulty to establish liver cancer organoid from patients, but with the experience acquired from this project, our co-PI, Dr. Stephanie Smart, has secured another research impact fund supported by RGC for the establishment of a liver cancer biobank from mice for translational liver cancer research, in which the liver cells in the mice will be genome edited by combination of driver mutations through this hydrodynamic tailwind injection that representing key molecular subtype of liver cancer. And then the tumor cell will be isolated from the xenograft to develop tumor organoids. And this biobank can be used for various types of study, including the co-culture, high-throughput drug screening, and multi-omic profiling. One unique features of our organoid compared to the 2D cell line is their dependency on the cell-cell and cell matrix adhesions. And previously, we have comprehensively studied these phenotypes on our cancer organoids by means of these ROCK inhibitors and spheroid formation assay, which we dissociate the organoid into single cell and subject them to the ROCK inhibitor withdrawal so that we can test the cell-cell adhesion dependence. We also put the cell in the ultra-low binding plate with Dalmatri gel so that we can assay the cell matrix adhesion dependence and withdraw ROCK inhibitor simultaneously, which we can test the cell-cell and cell matrix adhesion dependence simultaneously. And as expected, the normal organoids, they underwent apoptosis in all these 3 conditions, whereas some of the cancer organoids, they can survive in all these 3 conditions, showing this advanced phenotype just mimicking the cancer cells when they metastasize to the peritoneal cavities. And some of these cancer organoids show varying degrees of apoptosis in different conditions. And interestingly, this sort of in vitro cell-cell and cell matrix adhesion independency was actually associated with the in vivo tumor stage, lymph node and distant metastasis, as well as the infiltrative phenotype. Furthermore, the cell-cell and cell matrix adhesion independent signature score can predict both the overall and disease-free survival and associate with advanced stage in multiples of the public database in gastric cancer patients. We also established an xenograft model by orthotopic injections of our cancer organoids in the submucosa of the stomach, such that we can monitor the in vivo tumor growth by means of the bioluminescent signal. With this model, we are able to document the tumorigenicities of the RGAP fusion, in which its counterpart, when we lock out this RGAP fusion, can substantially inhibit xenograft formation. We also make use of our organoids, normal organoids, with the SCRIPA gene editing to understand individual driver mutation in terms of their molecular mechanism in driving gastric cancer. In essence, we engineer RHO-A and CDH1 mutations in the normal organoids, and we find that the RHO-A and CDH1 loss can confer cell-cell and cell matrix adhesion independency, respectively. To identify new vulnerabilities and to identify possible combination drug treatment in a high throughput manner, we have adopted CombiGem SCRIPA tools from our collaborators, Dr. Alan Wong, on our organoids. And he has also generated a newer version of CombiGem SCRIPA and a newer Cas9 that with a high fidelities and less off-target effects. And we have already adopted these new tools in our organoids to perform functional screen. Our co-PI, Dr. Jay Shi, also established a novel co-culture model between the tumor organoids and the NK cell, so as to understand the mechanisms for the immuno-oncology dynamics in 3D, which we hope to identify potential molecular mechanisms that can be targeted to develop new cancer immunotherapy. And in essence, the tumor organoids were engineered with the H2B that can mark the nucleus in red. And the primary NK cells were isolated from the human blood, were labeled in green. And they were co-cultured in the matrix gel and conduct 3D lifestyle imaging to look at their real-time interactions. And here, these are the representative videos that the NK cell co-culture with the organoids. On the left here is the MSI organoid, where on the right here is an intestinal organoid. As you can see, there are substantial NK cell movement and infiltration into the MSI organoid and lead to substantial cell death. However, there is a lot less of the NK cell motions and infiltration into an intestinal organoid. And we are trying to explore the underlying mechanism for these variabilities between NK cell and different types of gastric cancer, and hope to identify molecular mechanism that can be targeted to enhance the cancer response towards NK cell killing. Our co-PI, Dr. Zhang, has developed user-friendly and interactive web server called the SuperFastNet to understand the gene network and also the associated gene functions. So with just a few clicks on the web, we are able to, this web page can help to summarize the complicated biological pathway into this interactive eye-catching gene network. And this can further help to predict clinical response for those deregulated network. And he further developed another web server tools that help to characterize different immune elements, including the mutation burdens, tumor infiltrating lymphocytes, to classify cancer into either the immune hot or cold subtypes, and to understand the immunogenic and immunosuppressive pathway, which hopefully can help us to increase the understanding between the cancer and immune cell interactions and to give better predictions on the cancer immune treatment. To conclude here, we have established a primary gastric cancer organoid that include most of the subtype of stomach cancer. Our model capture regional heterogeneities and subclonal architectures that they look like and behave like a stomach tumor in the body, and enable large-scale drug screenings. And with the linked genomic data, it can provide useful resources for studying cancer biology and development of personalized cancer therapy. With our cancer organoid model, we are able to review critical growth characteristics that parallel aggressive growth patterns and advancing tumor stage in the patients. In our normal organoids, we are able to review new mutational signature through which are able to identify new carcinogenic agents. Through gene editing on organoids, we are able to reconstruct the pathway that drive cancer. And with the genome-wide screen, we are able to identify new therapeutic targets. And this TRS program has laid solid foundation for the widespread applications of the organoid technology to study other cancer types, and also with potential to study disease beyond cancer. And this project trained dozens of young professionals, including me, and students at the forefront of the genomic era. We all enjoy and proud to be in this project. And these are some of our joint publications arise from this project. And finally, on behalf of Professor Lang, I would like to express our sincere gratitude to UGC for supporting our research and advancing our goal towards the genome guide personalized treatment therapy to gastric cancer patients. And I also want to thank the co-PI, collaborators, colleagues, and students for your contributions in this project. Thank you very much.
Okay, thank you. Thank you, Dr. Yan. So if you have any questions, so please let us know. Yes.
Thank you, Helen, for the wonderful work and the beautiful talk. I have two questions here, two quick questions. The first question is that you, apart from the stomach cancer organoid, you mentioned that you already established NPC organoid. So to me, it's like NPC is a cancer with intensive lymphocyte infiltration. So I'm wondering whether there are any lymphocytes in your NPC organoid or only the epithelium component. The first question first, you may.
So as far as we know that this cancer organoid protocol only allows the survival of the cancer cells in the matrix gel. So theoretically, there should be no immune cell in the culture medium, in the culture conditions.
But my question is that are these lymphocytes can be passaged in your NPC?
No, so we're only able to see some other cell type like the fibroblasts, maybe some immune cell at the initial passage, but when we passage, eventually they just gone away. They cannot survive.
My question is, without lymphocyte, do you think is your NPC organoid still represent the native NPC?
Yes, this is also one of the so-called problems of the inherent defect of this model is just only allow the survival of the cancer cell, the pure cancer cell in the matrix gel. That's why currently a lot of researchers has been putting a lot of effort to develop sort of the co-culture model with other cell type with the cancer organoids.
Okay, I have a second question. You said you already established a drug screening platform.
Mm-hmm.
Could you please elaborate more on that? I mean, do you have publication for this?
Yes, yes, we have performed, as mentioned, we have already finished the drug screening on 66 tumor organoids on 37 drugs. But since we are really doing the manuscript preparations, and we also intend to apply patent on based on our discovery, therefore we cannot share our findings meanwhile.
Okay, we look forward to your publication. Yes. And thank you for helping us initially establishing intestinal organoid for our team. Thank you.
Thank you.
Okay, thank you. Any other questions from the audience? Yes.
Thank you for that presentation. I have a quick technical question. So when you make these organoids, the cells, you dissociate them to be single cell and then just put it in Metrogel? Is that what happens?
Yeah.
Okay.
So for normal, we use mechanical isolations as
Right,
we twist the tissue so that we allow the intact stomach gland to release from the tissue.
Right.
And then for the tumor, because of the stiffness of the tissue, so we need to have the enzyme digestions to
right,
make it become
okay.
single cell.
But the real question is about for the cancer, cells from the cancer samples. Do you purify the cancer cells? Because not every somatic cells have the mutation in some cancer tissues. So how do you? So when you do these cultures, do you do some cell surface sorting, or how do you do it?
Yeah, we try to get the organoids, the cancer organoid that really represents the real tissue.
Right.
So we try to capture all sort of cell that we can get from the isolation step. So
Right.
we didn't do any cell sorting on this.
So the final product, the organoid, though, may have a different composition of
Yes,
cells compared to the original tumor,
Right.
right?
Yeah.
So you think that's relevant, or that's not? It's not going to cause a problem for your screening.
Oh, I mean, we are not doing any sorting, so that we try to recapture everything that we can find in the real tissue.
But they change when you culture them.
As long as so far when we do the whole exome, the genomic and the transcriptomic profiling, as well as the epigenomic profiling, we see kind of really tight recapitulation between the organoids and the cancer tissue.
Okay, thanks.
Actually, I have a similar question. Actually, coming back to more basics, what's the definition of your organoid? And to me, if it's a clump of cells, it's a clump of cells. Okay, or you say an organoid must have some definition. What's an organoid?
So in the field of organoid, there are indeed two types of organoid. One is induced from the induced pluripotent stem cell, so usually linked to the normal organs. But another type is derived from the adult stem cell. So this is what we are using this protocol. So we just kind of collect the adult stem cell from the tissue and then supplement because of different leach factors that supports the growth of the stem cell in the tissue, so they can able to proliferate, divide in vitro continuously.
For me, an organoid must resemble an organ.
Yeah, so just as already published in the previous literatures by the inventor, Professor Hans Lab. So when they withdraw all these sort of leach factors, these adult stem cells start to divide, and they are actually able to give rise into various lineage that mimic the real organs. For instance, in the normal colon organoids, when they withdraw the wing, the Auschmundins, they are able to differentiate into the 4 essential lineage that commonly found in the normal colon.
Thank you for your explanation.
Thank you. Any other questions from the audience?
Maybe
Please,
I have a complementation to Helen's answer to Professor Chi's question. Basically, organoid is not just a cluster of cells. They can represent the genetic background, the cellular composition, the tissue architecture, and the functionality of the original tissue. So basically, that's the definition of organoid. Thank you.
Thank you for your supplement, Jay.
Have you tested them?
Verified
them?
Yes, we hope to.
Okay, any further questions for Dr. Yan? It's great discussion, so let's thank you, Dr. Yan.